practical histology. This laboratory about the cytology practical. We take the cytology theory. The cytology is the study of the structures of the cells which make up tissues and organs. In this laboratory, we deal the shape of the cells, which is the smallest unit of the living matter of the multicellular living organism. Each cell and the body differ in the shape, structure, and size. If take the structure of the cell consists of the nucleus, cytoplasm, organelles, and inclusion. We deal with the cells of the different types of the tissue, take the shape, structure. If we take any cells in the body consist of for example, these cells. This is the nerve cell. Nerve cell consists of the cell body, this cell body, and dendrite, axon, and dendrite. If we take the example of these cells is nerve cells, consist of nucleus and cytoplasm. This nucleus, nucleolus, and this basophilic area, the cytoplasm consists of what? Organelles, nasal substance. The nerve cell, this nerve cell consists of the nucleus and cytoplasm. The cytoplasm of the nerve cell filled with the different organelles, Golgi apparatus, mitochondria, centriole, inclusion, mitochondria. But in this slide, the nasal substance or the rough endoplasmic reticulum is very clear as basophilic area inside this cytoplasm. This blue area, all this blue area is the nasal substance or the rough endoplasmic reticulum. This slide take from what? From the nerve cell to observe very clearly which organelle is rough endoplasmic reticulum. Which name of the stain is used to demonstrate or to nasal substances hematoxylin and using the name of stain hematoxylin and using. This about the organelles rough endoplasmic reticulum inside the cytoplasm of what? Of the nerve cell. All this figure is nerve cell. Consists of what? Nucleus filled with the cytoplasm. This basophilic area or blue area, this nasal substance, rough endoplasmic reticulum. Another figure, also this nerve cell, this dendrite, axon, consists of the nucleus, this basophilic area, rough endoplasmic, which is very clear, nasal substance, hematoxylin, and using the name of stain. If we take another slide, this mitochondria, another organelle is inside the cell, mitochondria. To demonstrate the mitochondria, need a special stain. For example, genus green B. We need what? Special stain, genus green B. In this figure, we observe different type of the tubule because this slide or this section take from the what? From the kidney tubule. You know that the kidney tubule consists of the tubule. This tubule, this only also kidney tubule, this kidney tubule, also this kidney tubule. This slide consists of the different type of what? Of the kidney tubule. This glomerulus, this kidney tubule. Inside each cells of the kidney tubule, observed in this area. These cells inside the kidney tubule, also these cells consist of what? Organelles, nucleus, and cytoplasmic organelles. This organelle is mitochondria. We use it special stain, for example, genus green B to demonstrate what? Or to demonstrate the mitochondria. Inside this as what? As rod, which appear as what? Appear as rod. This slide or this section take from what? From the kidney tubule. This kidney tubule, all this kidney tubule. Inside each cells of the kidney tubule, there is cytoplasmic organelles, mitochondria. In this figure, the nucleus is not appear because we used the special stain genus green B only demonstrate the mitochondria. But if we, another slide, if we take the hematoxylin and using the nucleus is clear, cytoplasm is clear, but inside the, when use it genus green B or any special stain, the nucleus is not clear or not demonstrated. Only the one important point observed. Also, this figure demonstrated the mitochondria inside what? Inside each tubule. This all tubule. This tubule. Also, this tubule. Inside each cell of tubule, there is mitochondria in this shape. Inside each cells of the kidney tubule, there is cytoplasmic organelles called the mitochondria, use the special stain. Also in this area, as what? As a rod. Inside each cells of the kidney tubule, we know that the kidney tubule lined by what? Simple cuboidal epithelium. The cells appear at the pyramidal shape. Inside each cell, there is what? There is mitochondria, cytoplasmic inclusion. This mitochondria, this. Not all this, there is kidney tubule. Inside each, there is mitochondria. This kidney tubule. Inside each cell, there is mitochondria. As what? As rod. This mitochondria in each cell. Another slide or another organelles inside these cells. There is Golgi apparatus. Also, this organelles observed by using special stain silver nitrate. Silver nitrate. This slide take from what? Take from the epididymis. Epididymis, tubules of the male reproductive system, take from what? Epididymis. This cells, this all section from what? From the epididymis. Inside each cells, there is cells and organelles. Also, this Golgi apparatus appear as what? As the positive Golgi image. This black area Golgi apparatus, positive Golgi image. This black area, this Golgi apparatus as positive Golgi image. Also this positive Golgi image. Arrow demonstrate or indicated the positive Golgi image. This positive Golgi image. Inside each cells which line the epididymis. This cell, this cell, this cell. Different type of cells. Each cell consists of the kidney tubule, appear as the positive Golgi image inside the cells which stained by special stain silver nitrate. This positive Golgi image. This is black network. The arrow, positive Golgi image inside each cells which line the kidney, the epididymis. Also, this slide appear the positive Golgi image inside also the kidney tubule. This black network positive Golgi image. Which type of this stain? Silver nitrate. Positive Golgi image, silver nitrate. Also, kidney tubule. This kidney tubule. Kidney tubule. Kidney tubule. Inside each cell, there is a nucleus organelles. This organelles positive Golgi image. While if we use the hematoxylin and eosine, for example, taking the connective tissue plasma cell, there is Golgi apparatus appear as the negative Golgi image inside the cytoplasm of the plasma cell as what? As negative. But if we use this special stain silver nitrate appear as the positive Golgi image. In the hematoxylin and eosine, negative Golgi image. But if we use by the special stain like silver nitrate, positive Golgi image. This slide or this figure shows the cytoplasmic inclusion. For example, lipid droplet or fat cell. To demonstrate the fat cell, we use the special stain silver nitrate or osmic acid. This lipid droplet. Lipid droplet. Drop. Black appear as what? Black area. This lipid droplet. This section take from the adipose tissue, fat inside the body, to demonstrate the inclusion inside the any cells of the body. For example, lipid droplet, we use the special stain adipose tissue, or name of the stain special, the stain silver nitrate or osmic acid. Another slide to demonstrate the inclusion or cytoplasmic inclusion inside the cell. There is glycogen granule. The glycogen granule occur inside which type of cell? Hepatocyte, or another name, liver cell. If we take some note or some information about the hepatocyte or liver cell, the hepatocyte have six side or hexagonal shape. Consists of two nucleus, binucleated. Number one, number two nucleated. We know all type of the cells in the body consists of the one nucleus, but liver cell consists of two nucleus. Observe this two nucleus. This also two nucleus. But all type cells in the body consist of only one nucleus, except the liver cell consists of the two nucleus. Cells of the bone osteoclast, for example, consist of the more than 2 or 3 nucleus, multinucleated, but hepatocyte binucleated, two nuclei, binucleated, two nuclei. This about the shape and some information about the liver cell. The cytoplasm of this type of cell or the liver cell filled with the cytoplasmic inclusion, for example, glycogen, this red area, this glycogen granule. Cytoplasm of the liver cell filled with the glycogen granule. This name of a stain to demonstrate this inclusion, Pestis carmine. We uses the Pestis carmine stain. This slide take from what? From the liver cell or hepatocyte to demonstrate the cytoplasmic inclusion, glycogen granule. To demonstrate the what? The cytoplasmic inclusion, glycogen granule. This red area, cytoplasmic inclusion. This cells consists of the nucleus and cytoplasm. Nucleus, cytoplasm. The cytoplasm filled with organelles inclusion. This slide demonstrates or explains the cytoplasmic inclusion, glycogen granule inside the liver cell. This glycogen granule. Also in this area is very clear, this glycogen granule, this nucleus, this glycogen granule. Nucleus, glycogen granule. Nucleus, glycogen granule. We use it, the special stain. The name of stain is this carmine. In this laboratory about the cytology, the name of stain is very important. The name of stain is very important. Very important, name of stain. This about all the information about the cell.